Hi guys, welcome to the first lesson in our hormonal regulation of metabolism section of the course, at least in terms of the physiology. A couple of preliminary things to talk about. First of all, I will talk a little bit about insulin and glucagon, but for the most part I'm going to be concentrating on the other hormones that are involved in this regulation. The biochemists will be talking to you about the insulin and glucagon and their major effects. And then I'm going to come back and give you a couple of different scenarios, exercise and diabetes, where these hormones are important as a whole and the way that they interact. So I'm going to be going over the hormones of the adrenal gland, which is cortisol and the catecholamines. I'm going to be going over thyroid hormone and growth hormone. And then I'm going to introduce the topic of insulin and glucagon. And biochemistry is then going to talk to you in more depth about insulin and glucagon and the feed-starve cycle. And then I will come back and cover exercise and wrap up with a lesson on diabetes and what happens with these hormones there. Although I'm going to be talking a little bit about diabetes as we go along. It's a very important disease and it's hard for me not to talk about it when it comes to regulation of glucose. All right, so we're going to start with the hormones of the adrenal gland. That'll be cortisol, and then the next lesson will be about catecholamines, which are epinephrine and norepinephrine. Hi guys. So what follows here is a very, very brief review of the effects of cortisol on or some of the non-metabolic effects of cortisol. I recorded this before we added lessons on cortisol to the HDHR block and the vital fluids block. So double time your way through this. I don't plan to concentrate on this material on the exam, but I do expect you to remember it and know it, so it will be worth your time just to review it. And that does include the feedback loops, which I'm going to conclude with another little inserted recording, desktop recording. So if you're not double timing your way through this, go ahead and double time your way through all the way up to the feedback loops and just briefly review it. And when we talk about cortisol, which is the topic of this lesson, we are talking about secretion from the adrenal cortex. Now, your adrenal gland mediates several physiological functions, many of which you are already aware of from previous blocks. What we're going to concentrate on, the gland is important in controlling metabolism and responses to injury and stress. Stress is something that we'll be going over later on in the curriculum. We'll be talking about a lot about how these hormones interact in response to that. Okay. When we talk about blood metabolism, we are largely talking about cortisol, although not exclusively. All these hormones have effects on metabolism. And when we talk about the physiological functions mediated by the adrenal gland, control of blood glucose is the one that's of central importance here. We'll talk about protein turnover and the effects of cortisol on that. Cortisol is kind of unique in that respect. We'll talk about fat metabolism a little bit, but blood glucose regulation is what this is really centered on. In the same way that, and you're going to find out there's a lot of similarity here, in the same way that pressure regulation is the center in the cardiovascular portion of the curriculum. Okay? The adrenal gland, important in electrolyte balance. You know this. We've talked about aldosterone. We talked about the kidney and its effects on sodium reabsorption, which it increases, and potassium secretion, which it also increases, and upon calcium as well. All right. So aldosterone we've covered a great deal already, but you are of course aware of its secretion from the adrenals. And then we've talked a little bit about the catecholamines as well. And the fact that epinephrine is secreted from the adrenal medulla, and it regulates cardiovascular tone. It's involved in stress as well. But we've talked a great deal about norepinephrine and epinephrine and its cardiovascular effects, okay? So this is not a histology lesson, and I'm not going to pretend to be a histologist, but just to make sure we're all up to speed. The adrenal separated into two parts, the cortex and the medulla. The medulla secretes your catecholamines. The cortex, specifically the fasciculata, secretes the cortisol. And that is about all you are going to hear from me about this organ in terms of the histology. Okay, so here we have a measure of cortisol levels over a 24-hour period, okay? And what you can see, first of all, is that the cortisol is highest in the morning. It actually rises as you sleep and is highest at the morning, and it may be one of the things that kind of wakes you up in the morning and determines that you get up, okay? The second thing that you'll notice is that it's pulsatile. That is, it isn't like a just steady decline and steady increase. You get these pulses of release. And the reason for that is because the ACTH is released in a pulsatile manner. So if you look at the release of ACTH, here we have ACTH on the y-axis and the same x-axis, the 24-hour cycle here, you'll notice that the cortisol follows the ACTH. The ACTH goes down during the day, rises at night, and peaks during the morning, and it also is released in this pulsatile fashion. So the cortisol parallels that. It's highest in the morning and lowest when you go to bed at night, and it also is released in a pulsatile manner, okay? And here is another figure that sort of demonstrates that. This is the average of many individuals. So what that has done is it's taken a lot of the pulses out. And so what you get is more of a smooth curve where you can more easily see the cortisol rising during the sleep cycle and peaking in the morning, and then slowly declining during the day, okay? All right. So in terms of the characteristics of cortisol, we finally get to that. Obviously, released from the adrenal gland, it is a steroid hormone, a derivative of cholesterol. It is released in response to ACTH, you now know that. Like most steroid hormones, virtually all of them, it is bound to plasma proteins, and that is the way that it's transported in the blood. We have a cortisol binding globulin in particular, that's worth noting. It acts by entering across the membrane because it's nice and lipophilic, and it enters the nucleus and alters gene transcription and enzyme production, so that's the way that it mediates its effects. And what we're going to talk about mostly here is its participation in control of metabolism, mostly control of substrate levels in your plasma. Okay, so very briefly, let's review the feedback loops for this organ, and I do expect you guys to know this and be able to manipulate it. So if you need to, go back and review the notes from Vital Fluids. I don't think you'll need to. So you guys will recall we're dealing with three structures here when it comes to regulation of the secretion of cortisol. We have the hypothalamus. That's connected to the anterior pituitary via a portal system here that carries hormones from the hypothalamus directly to the organ. And then we are dealing with the target organ in this hypothalamic pituitary target organ axis, and in this case it is the adrenal cortex. The hypothalamus secretes CRH, corticotropin releasing hormone, and again this goes through the bloodstream, down the stalk between the hypothalamus and the ventral part of the brain to the anterior pituitary, and CRH stimulates the release of ACTH from the anterior pituitary, and the ACTH goes into the general systemic bloodstream and finds its way to the adrenal cortex, where it stimulates the release of cortisol. Now, the feedback loops, again, to remember and concentrate on, we have the feedback of ACTH back upon the hypothalamus. And this is a negative feedback loop. So this is a control mechanism by which ACTH controls its own release. If the ACTH gets, if you increase the release of ACTH, it still goes up, but this feedback loop limits the increase, and so it keeps that secretion under control. And the same thing is true of cortisol, which feeds back upon the anterior pituitary and the hypothalamus both to decrease the release of ACTH and CRH. So again, we're dealing with a negative feedback loop here, where cortisol is controlling its own release. If you have a condition like hypoglycemia that stimulates the release of cortisol, the cortisol will still be up at steady state. It's just that it won't be up as high as you would expect due to the existence of these feedback loops, which keep the release of cortisol under control, so that none of these one mechanisms can completely dominate the release of cortisol. You have hypoglycemia, you want to increase your cortisol release, but you don't want to do it in such a way that you mess up any of the other things that are sort of under the control of cortisol, which you don't want to get out of control just because the hypoglycemia was there. So again, that's a very brief review of the feedback loops and the hypothalamic pituitary target organ system axis. And again, if you really feel like unsteady on the details of this, then you should go back into the vital fluids material and review it. But otherwise, just remember it. I do expect you to remember it. All right. In that respect, the control of metabolism takes place very similarly to the way that your body controls blood pressure in the cardiovascular system. In the cardiovascular system, your body does what it takes, usually through the sympathetic and parasympathetic nervous systems, to try to keep blood pressure high. And you know that. Your body assumes, as long as blood pressure is high, that your organs can be perfused. It's smart enough to know that if your blood pressure is not high, it doesn't matter what you do on the organ level, you're not going to get a perfusion because you need a pressure gradient in order to perfuse the organs. So your body concentrates on keeping blood pressure as constant as it can. Okay? In the same way, your body concentrates on keeping glucose levels as constant as it can. Glucose is your regulated variable in terms of metabolism. Okay? If glucose gets too high, your body will react in such a way that it will bring it down. And I will tell you that the primary mechanism by which it does that is by stimulating the release of insulin, which we talked to you about a great deal. Okay? If your glucose levels get too low, then your body will do what it takes to get the glucose levels back up, because it knows that if the plasma substrate is too low, it doesn't matter what else it does, your tissues aren't going to get enough glucose to sustain metabolism, okay? So glucose, plasma glucose, is your regulated variable, all right? Okay. What you're going to find is that, again, very similar to the cardiovascular system, there are going to be local mechanisms that are going to be in play to make sure that your tissues get an adequate amount of glucose where it's needed, okay? So there's going to be, and cortisol is going to be an example of this, a certain amount of what may not on the surface make a lot of sense, okay? You remember we talk about exercise and we talk about the fact that you get an increase in sympathetic nervous system activity, and this causes vasoconstriction, and the question always comes up, why would you want to vasoconstrict to skeletal muscle when you're exercising? You want your skeletal muscle to get more blood. And the answer is that there are local mechanisms in place to make sure that vasodilation takes place in the active muscle so that you get increased perfusion where it's needed. That's going to be what's going to take place here. You're going to be doing things like inhibiting glucose transport in order to sustain glucose levels in the plasma. And when I tell you that, you're going to have to bear in mind that there are other local mechanisms, and I'll demonstrate at least one when we talk about exercise, that will allow adequate uptake of glucose to take place in tissues where it's needed, okay? So sustaining glucose levels is priority number one on a systemic level, and then locally what you're going to find out is that there are going to be ways to make sure that glucose uptake continues to take place into tissues like skeletal muscle where it's needed, all right? All right, so bear that in mind. Glucose regulated, your body on a systemic level, that means an endocrine level, will do what it takes to sustain glucose levels and regulate them. And then on a local level, you will be able to maintain glucose uptake and continue to get glucose into your cells when it's needed, all right? All right. This is almost all of the hormones that we're going to talk about when we talk about control of metabolism. The only one missing is thyroid hormone, okay? So here we have a variety of different biochemical reactions that regulate blood glucose and blood fatty acids and amino acids, all right? And here we have all of our hormones listed. D means down, I means increased, N means no change, okay? All right, so the first thing I want you to notice is what insulin does, okay? Again, the biochemist will talk to you in detail about insulin. But insulin is secreted when blood glucose levels get too high. And what you're going to find out is that virtually all of its actions are targeted to getting that blood glucose back down toward normal, okay? So you are going to decrease glycogenolysis. You're not going to want to break down glycogen when your plasma glucose levels are too high, because increasing breakdown of glycogen is just going to increase the glucose levels more. You want to decrease glycogenolysis. You decrease gluconeogenesis. Who wants to make new glucose when the glucose levels in the plasma are already high, okay? You will, in fact, increase glucose uptake with insulin, okay? You will decrease blood glucose. That is insulin's primary purpose, to decrease plasma glucose. You will decrease lipolysis, because your body figures if you are, and you will decrease blood fatty acids. So assuming that your blood glucose went up in response to eating a meal, then you aren't going to want to increase blood fatty acids any more than you're going to want to increase blood glucose. Your plasma has plenty of substrate. You aren't going to want to increase the blood fatty acids to supply more substrate. That's not needed here. So we get a decrease in lipolysis, and we get a decrease in proteolysis, and a decrease in blood amino acids. As you guys know, these can be converted to energy as well, okay? And so that isn't something that you want. So insulin's targets here, its effects here, are all aimed toward getting substrate in the plasma down, okay? Or at least not increasing it, okay? So insulin would be something that you would secrete in response to a meal, all right? On the other hand, these four hormones are all secreted to do exactly the opposite, okay? In general, these hormones are secreted when blood glucose gets low, and their primary purpose is to increase blood glucose, okay? And bring it back down toward normal. So I think just looking at this chart, you can tell why diabetes is such a problem. We have all kinds of redundancy on this side. If something goes wrong with one of these hormones, there are other hormones that pick up the slack. The effects on metabolism won't be disastrous, okay? I'm not saying there won't be effects, but it isn't like what will happen if insulin goes away. If insulin goes away, there is nothing left to bring blood glucose levels down, okay? And what happens is these hormones go wild and they dominate, and what happens is you get high blood glucose and you can't bring it down, because the only way to bring it down is through insulin. And if you don't have insulin, there's nothing to back it up. These hormones individually all have backups. This one does not, okay? So this is really, it's the primary point of the diabetes lesson, okay? This is insulin, it's all alone, okay? And if it goes away, there ain't nothing left, okay? So having said that about these hormones, let's talk a little bit about cortisol, all right? Cortisol's general effect is going to be to increase substrate levels in the blood. And I say general, okay? It will increase gluconeogenesis, okay? And that should make sense to you. You're trying to bring plasma glucose up. It will block glucose uptake in the tissues. This will stop glucose from disappearing from the plasma and maintain plasma glucose. Now bear in mind, you're blocking glucose uptake, and I know it makes no sense because the primary reason why you want to sustain blood glucose is to make sure that tissues that need the energy will get it. They will, okay? Despite this blockage, tissues that need glucose will get the glucose that they need, okay? The primary purpose of cortisol is to keep the plasma levels up, because if the plasma level's up, it's not going to matter what you do to glucose uptake. There's no glucose there to take up. So cortisol blocks glucose uptake to keep the glucose levels high, or at least bring them back up toward normal, and therefore you get an increase in blood glucose levels. You get an increase in lipolysis and an increase in blood fatty acids, okay? And this again makes sense to you. You want to increase substrate so that it can be metabolized by the tissues in a time of need, okay? Unique amongst these four hormones here, cortisol actually increases proteolysis and increases blood amino acids. The other ones don't, and this is the primary reason why you get an increase in amino acids in response to stress, okay? These other hormones, cortisol, as you will eventually find out later in the curriculum, is one of your primary mediators of the stress response. And so what you're going to find out is that blood amino acid levels go up, and that's primarily because cortisol goes up in response to stress, okay? So this is unique. It's something to remember. It's unique amongst these four hormones. It's the one that mediates the increase in amino acids. The other thing that's unique about cortisol, it decreases glycogenolysis. You would ordinarily expect that to increase like these other hormones, but cortisol does not do that. It decreases it. Okay, so that's kind of unique in comparison to those other three that you see. So generally speaking, cortisol increases substrate levels in the plasma. It uniquely increases amino acids, and it uniquely decreases glycogenolysis. Okay? All right. So here we have exactly what I talked about, your effects of cortisol. You increase liver glycogen stores, okay? You raise the amount of utilizable substrate, particularly blood glucose. You do that by decreasing the effect of insulin on the tissues, okay? And that tends to decrease glucose uptake and maintain plasma glucose levels, bring them back up toward normal, just like you see here, okay? Cortisol increases blood glucose back toward normal. It does that in part by decreasing glucose uptake and by doing things like increasing gluconeogenesis, okay? All right. And here is the effect on amino acids and protein metabolism. Here we have the plasma concentration of these amino acids and the blood production rate of these amino acids, okay? And you will note that in response to cortisol, the production of leucine goes up and the plasma level of leucine goes up in response. The production of isoleucine and valine also go up. This is because you've increased proteolysis, okay? Alanine is a little bit unique here. You're producing more alanine through proteolysis, but in fact, the levels stay relatively constant, and the reason is because you're using alanine for gluconeogenesis, and so you're actually using that up and preventing the plasma levels from accumulating, unlike these, okay? All right, so that's all I've got on cortisol. Thank you for your attention.